To consider the ways that we regulate cardiovascular function, we're going to concentrate in this section on the medullary cardiovascular center, referring to a part of the medulla in the brainstem that controls cardiac function. In considering cardiac output, remember that there were two components. There was stroke volume and heart rate. And so now we want to consider what regulates that heart rate. And it's a region in the medulla in the brainstem called the cardiovascular center. The cardiovascular center in the medulla is actually made up of two different regions that aren't mentioned here, but they are the cardioacceleratory and the cardioinhibitory areas. So it has the ability to both increase and decrease heart rate and therefore cardiac output. Now, the inputs to this area come from a couple of different regions. First off, higher centers in the brain, and the cortex and the hypothalamus and those sorts of areas can send input down to the medulla to modulate what's happening in the cardiovascular center. Also, there are some reflexive inputs that come through sensory connections to this area. When we change body position, when we sit up from lying down or stand up from either of those positions, the cardiovascular center has to increase the flow of blood to the brain to compensate for the change in gravity's effect on blood. There are chemical changes happening in the blood, whether it's pH or CO2 or oxygen levels, those sorts of things, which need to be regulated. And then this blood pressure changes as there's stretch within the walls of blood vessels or in the wall of the heart. There will be input to the cardiovascular center. Any of those inputs will cause the kind of changes that we're going to see, but they can come from any of those sorts of places. This is an anatomical representation of the connections from the cardiovascular center out to the heart. Now, it's very important that you understand to begin with that the connections coming out of the medulla here are not anatomically correct representations. The vagus nerve is the preganglionic fiber coming from the brainstem for the parasympathetic division. The dot that we see here that has a synapse from a little yellow input would actually be in the brainstem. Now, there's a neuron from the cardiovascular center to the vagus nerve nucleus, which is represented, but that would really be internal to the brainstem. And then that preganglionic fiber would come out of the brainstem, go to the heart, synapse on the terminal ganglion there, which is represented by the second purple neuron in the pathway. And then also the sympathetic system is a little bit more complex than suggested here. The yellow line that we see coming out of the cardiovascular center would actually descend through the brainstem and into the spinal cord to synapse on the preganglionic neuron in the thoracic spinal cord. That would then emerge from the spinal cord and synapse in the sympathetic chain ganglion at that level or possibly at a more superior level in the chain. And then the fiber that we see here labeled the sympathetic cardiac nerve would be the postganglionic fiber coming from that. So just to be sure, the anatomy that we see here is a little bit simplified for the sake of the picture, but it would actually be mostly internal. The yellow projections that we see here would be internal to the central nervous system. Now, the other thing I want to point out here is the parasympathetic input, the vagus nerve and the terminal ganglion, that is its target, is only contacting the SA node and the AV node, which are the two targets that we see there at the end. The cardiac nerves from the sympathetic side go to the SA node and the AV node also, but also have a branch that goes to the ventricular wall in general. The SA node, AV node targets, those are to regulate heart rate. The one from the sympathetic system going to the ventricular wall will be able to affect contractility, which is to increase calcium influx. This figure has been modified slightly from the book's version. The label for each of the three frames here has been changed to specify what really should be represented here, and the time frame on the x-axis has been altered. The first frame is showing us the pacemaker potential that we discussed earlier for the SA node. Fires off at a given rate and slowly reaches threshold between each spike, causing the next one to automatically fire off. Now, when we looked at this for the SA node before, the time between two spikes, two action potentials, was about 0.8 of a second, which would be 75 action potentials per minute. Here I've changed the time frame to be more accurate, representing it as about 0.6 of a second delay between action potentials, which would be about 100 beats per minute, which is what the intrinsic rate of the SA node would be, firing on its own without any autonomic input. Then the next frame again has been changed to represent a different rate, and this rate, based on the time frame that I've changed on the x-axis, would be between 60 and 80 beats per minute. I would have liked to have made it be exactly 75 beats per minute, which is the resting heart rate that we usually talk about, but the way that it's drawn, it's not exactly 75 beats per minute, but it's in that range of 60 to 80 beats per minute. And that's what happens when the parasympathetic system has an effect. It hyperpolarizes the cell. So instead of the lowest point for the pacemaker potential being at about -60, it's lowered, closer to -70. So it takes longer for the cell to get up to threshold. And then in the final one, we have an elevated heart rate of about 150 beats per minute. Again, it's not exact, but based on how I changed the time frame on the x-axis, it's a fairly representative heart rate here. What happens here is that the sympathetic input is hyperpolarizing the membrane of the cardiac cell. And so instead of going down to -60 between beats and then having to spend some time having to spend some time depolarizing the threshold, the distance to threshold is shorter, and therefore the time between action potentials is also shorter. Again, we have a summary figure that puts all of this together. As we were talking about cardiac output in the last section, it's a combination of heart rate and stroke volume. Stroke volume is the difference between end diastolic volume and end systolic volume. End diastolic volume is largely the result of preload, although that can also affect end systolic volume. And systolic volume is really the effect of contractility, which can be adversely affected as well through afterload. Preload is really about how much blood is returning through the venous system to the atrium and how long that filling can take. Contractility is really under autonomic innervation control, as well as some hormones that can have an effect. And then afterload, in an acute sense, can be controlled by vasodilation or vasoconstriction, but again, chronic high blood pressure is not something we can actually deal with. We'll be talking about venous return and vasodilation and vasoconstriction in the next chapter on blood vessels. And then filling time is really just a matter of heart rate, autonomic innervation we've talked about, and hormones I've mentioned where they play a part too. Heart rate is really just affected by autonomic innervation or hormones, again, which we just saw for contractility, although the autonomic innervation here includes more of the parasympathetic input to keep heart rate low for the resting level. The atrial reflex is really something kind of tied into venous return. As the atrial wall stretches, it's going to, through a short reflex, do something to the autonomic control of heart rate, trying to keep the blood from filling up the atrium too much. And we'll actually see something similar to that when we consider the hormonal control of blood pressure in actually the renal system. That'll come up. We can kind of see how a lot of things affect this. We'll be talking more about some of these in the next chapter, and some of these are just extensions of what we've been talking about with the autonomic and endocrine systems. Consider this question. When you feel that you've reached an answer, hit the next button to go into the next slide, which will reveal the correct answer. This question is actually trying to get you to think about the anatomical incorrectness of that figure that I showed you earlier. The cell body that causes norepinephrine to be released onto the SA node would be a postganglionic neuron of the sympathetic division. Norepinephrine, sympathetic neurotransmitter, and the postsympathetic fiber is going to end on the SA node. So being sympathetic and being postganglionic, it has to come from the chain ganglion. In other circumstances, it could come from a different ganglion, but for the heart, definitely it's covered by the chain ganglion.